So, hi everybody, welcome back after the coffee break. Now we are going to start a very exciting second part of this symposium. And the first speaker is going to be Florian Lempp from Biotechnology. And the title of the presentation will be VIR-2218, an investigational RNA interference therapeutic targeting HBV infection. Or you change it then. No, it's fine. Florian, please, go ahead.
Thank you, Maura. Good morning, everyone. My name is Florian Lempp. I'm a director for virology at Biotech in San Francisco, and I'm happy to give you an overview and an update on our RNAi program, VIR-2218. I'll start with some preclinical characterization and then very quickly move into a quick overview of the current clinical data. So VIR-2218 is an siRNA targeting the HBx open reading frame. We've heard that earlier today where all the transcripts of HBV overlap and has the potential to knock down all transcripts, both from cccDNA as well as from integrants, because it targets upstream of the major integration breakpoint. It's GalNAc conjugated for efficient delivery to the liver, and it has Alnylam's ESC+ technology for improved safety and reduced off-target activity. In preclinical studies that was shown on a poster earlier on the conference, we see very potent knockdown in, for example, PHH in vitro infection models with double-digit picomolar activity. We see pan-genotypic activity across different genotypes and different isolates of genotypes, and we see potent knockdown also in integrated cell lines. VIR-2218 is in several clinical trials currently in parallel. I listed them out here. In the 1001 study, part A, we are investigating VIR-2218 monotherapy. In the part F, we are investigating 2218 plus PEG interferon combination therapy. In the March trial, we're investigating 2218 plus our monoclonal antibody VIR-3434. And in some cohorts with interferon alpha, all of this is in virally suppressed patients. Then in the PREVAIL study that just started a couple of weeks ago, we're investigating the same agents but in immune active and inactive patients. And in the SOLSTICE trial, as shown during the conference, we're investigating 2218 and 3434 in HDV co-infected patients. And then on top of that, we have several collaborator-led studies where we're investigating different combinations with collaborative agents like TLR8 agonists, for example. So today in my talk, due to time restrictions, I'll only focus on these three cohorts up here where we have data on virally suppressed patients. If we just look at the mode of action of these different drugs, 2218, as said, silences all HBV RNAs, inhibits intracellular replication of HBV, PEG interferon alpha. We all know it is an immunostimulatory agent, and then VIR-3434 is our monoclonal antibody targeting HBs antigen, which can inhibit viral entry. It can bind to and eliminate subviral particles and virions from circulation, and then has the potential through Fc gamma receptor, Fc-dependent mutations that enhance Fc gamma receptor interactions to lead to stimulation of T cells and a potential vaccinal effect. Starting out with 2218 monotherapy, in this trial that I'm showing you here, the 1001 part A, we were looking at two different dose regimens at 200 milligram once monthly doses, either 2 doses or 6 doses. No major ALT elevations were observed. The drug was generally safe and well tolerated. A couple of few grade 1 ALT elevations was the only thing that was recognized. If we look at efficacy, we're looking here at an HBs antigen plot here. As you can appreciate, there is a reduction of HBs antigen with both dose regimens, leading about to 1.5 log to 2 log reduction at the nadir. And then upon removal of the drug, there is a rebound of viral antigen, HBs antigen, while with the 6-dose regimen, that reduction is more sustained. And also, if we look at the single patient data here, you can also appreciate that with the 6-dose regimen, we have these two cohorts of patients here, one's in green and one's in black. The green ones have a more sustained response even after withdrawal of the drug, whereas the black patients have faster rebound. Looking at the combination data of 2218 plus PEG interferon alpha, and this is a very busy slide. I tried to put all the most important data onto one slide. This was presented at EASL just earlier this year. So these are a couple of different cohorts that I will walk you through. Cohort 1 is 2218 mono treatment. Cohort 2 and cohort 3 are combinations with PEG interferon alpha, 12 and 24 weeks of PEG interferon alpha, and then cohort 4 and cohort 5 are really long-term interferon alpha treatment up to 48 weeks. You can just see at the HBs antigen reduction plot that we see the initial decrease in all cohorts similar to the mono treatment, which is here in orange. And then in the longer interferon alpha treatment regimens, we see that more sustained HBs antigen decrease, especially in cohort 5, that really has the longest interferon treatment. And then if we look at HBs antigen loss and seroconversion, which is our major endpoint, here at end of treatment, listed down here in this table, you can see in cohort 1, monotherapy 2218 only, there's not a single participant with HBs antigen seroconversion. With 12 weeks interferon alpha and also 24 weeks, we have one patient each in each cohort. But then in the longer treatment, up to 48 weeks interferon alpha, we have 4 or 20 to 30% HBs antigen seroconversion. And then if we look after treatment at end of follow-up, 24 weeks post end of treatment, 3 or 2, which is respectively 15 to 16% of patients remain HBs antigen seroclearance. And we found a factor that may be predictive of sustained seroclearance, which is anti-HBs at the end of treatment, which you can see in this graph up here. Those patients that rebounded after end of treatment had at the end of treatment anti-HBs titers of less than 500 milliIUs per mil, whereas those patients that had sustained HBs antigen seroconversion had titers higher than 500 milliIU per mil. So this data is very interesting. I want to point out that these cohort sizes are very, very small, 13 and 18 cohorts. Also, the numbers of patients with seroconversion are rather small, 4 patients each. So I don't want to overinterpret that data, just showing that it's certainly going in the direction that we want it to go. And then lastly, the combination siRNA 2218 with our antibody 3434. Here in this March trial, this is the first data cut from the March trial, we see different treatment regimens where we either have a lead-in with siRNA and then come in with the antibody, or we start both at the same time. If we maybe start with both at the same time, you can appreciate the fast and sharp drop in HBs antigen, which is mediated by the antibody via 3434. And then we have this continuous slower decline, which is mostly mediated by the siRNA. Upon removal of the drug, we see the rebound. And the same is seen if we have the siRNA lead-in. You see the slow reduction of HBs antigen mediated by siRNA, and then once the antibody is added, it leads to a sharp drop of HBs antigen with the subsequent rebound. But the rebound is not back to baseline, actually, but it remains, or it plateaus out as about 1 log reduction. In this trial, no participant has been found with HBs antigen seroconversion. Summing up, VIR 2218 is an investigational RNAi therapeutic that targets the HBX region to silence all of the HBV transcripts. It shows potent pan-genotypic antiviral activity in vitro models. And in clinical trials, HBV VIR 2218 is very well tolerated so far. Treatment leads to a sustained decrease of HBs antigen and can lead to seroconversions when combined with PEG interferon alpha. And currently, we're looking at the triple combinations of siRNA plus antibody plus PEG interferon alpha for up to 48 weeks in different cohorts, both nuke suppressed as well as the immune active patients that I mentioned earlier. Okay, I believe I'm not taking any questions right now, so thanks for the attention and happy to discuss later in the roundtable.